So without further ado, I want to introduce Dr. Ross Levine from the Memorial Sloan Kettering Cancer Center. He is the chief of the Molecular Cancer Medicine Service at HOP, the Human Oncology and Pathogenesis Program. Dr. Levine is a world-renowned physician scientist in myeloid malignancies, and he sits on the Research Oversight Committee for the Leukemia and Lymphoma Society and is one of the leads for the BEAT AML Master clinical trial. Ross, thank you so much for joining us, and welcome to the virtual event today.
Thanks, Hannah. It's great to be here today and to be able to present some of the work that a group of us have done together, both here at Sloan and our collaborators at a number of places, including lots of great collaboration with the folks at Mission Bio. And here are my disclosures, and to make a point that I did just recently agree to help advise Mission Bio itself. So I just put that disclosure there, although everything I'll talk about today is academic work from my lab done in academic collaboration with them and everybody else. And so just a few words. Obviously, you heard today from Hannah that it's AML Awareness Day. I have to say in full candor, I wasn't aware till today that there was such a thing as AML World Awareness Day, despite more than 15 years taking care of AML patients. But obviously, to me, every day is a day to think about AML patients, and it's great to be here with all of you to think about some of the biology. But before we talk about that, I just want to really accentuate that this is a challenging moment, and it's a challenging moment for all of us. And fundamentally, what we need to do is to realize that we can only do the best we can, and we can only do that together. So for young investigators, we understand how hard this is. We realize it's challenging, and we're going to do everything we can to support you. To our colleagues who are on the wards taking care of patients, especially those that have been pressed into service to take care of patients in this COVID crisis, thank you. And please stay safe wherever you are. Don't worry if you can't get lots of work done. It's a challenging time. It's hard to do work in the best of times at home and harder in this landscape. And try to stay connected with your community. And then two little, I guess, things that I try to do. One is focus on little things. Try and get something done in discrete units. It's all I can do. That big project's hard to do, but if you break it into chunks, you can do it. And find new ways to contribute. You know, review grants and papers. Talk to a new collaborator. Go on online and listen to lectures, not just in your area. Like if you're an AML person, it's great you're here, but go and hear about science and computational genomics or something you don't hear about. You know, get outside your comfort zone and expand your horizons. So I really encourage all of you to do that. But today is about AML, and I do take care of AML patients. And I think it's important to recognize that AML has really been a challenge for us, both biologically and clinically. On the left, I think you can see that the survival of AML has not increased substantively, and we've only really had a small number of therapies approved for AML over the past 4 decades. It's gotten a little better in the last year or two, but it remains a difficult disease to treat and a very difficult disease for families and patients to deal with. And the other challenge is that AML is the disease of the aging population, and we can't forget that the story of AML is not just of young children and adults with AML, but the median age is 68. And so it's a challenge to take care of older adults with AML who have many other comorbid conditions, and it can be challenging, and the disease is more complicated. And then on the right, this is just gene expression profiling, looking at bulk gene expression from about 500 AML patients. And it makes the point that AML on an inter-patient basis is incredibly complex. It's not one disease, but many. And as you'll hear today about single cell approaches, it's clear that in every patient, there's many different biological processes and clones going on. And this sort of earlier analysis doesn't capture that. But on the other hand, AML has some aspects that make it tractable. And that is that there are only a small number of recurrently mutated genes, probably 50 to 70, and only 3 to 8 coding mutations per AML genome, at least in bulk. And what that means is that you get a lot of biological complexity between patients, and I'll show you in patients, based on a small number of somatic events giving rise to mutational diversity. And that really suggests that there's a smaller number of events in the palette of possibilities for AML cells to sample from. But as we'll talk about today, it can do so in many different orders and permutations and in parallel. And we need to understand that process. This data from TCGA was a great look at the bulk genomic landscape. But the question is, how do we go beyond that? And we've learned in the last few years that AML is not a entity that arises de novo from normal stem cells, but rather evolves in a sort of accumulative route from a precursor condition like clonal hematopoiesis or chronic myeloid diseases like MDS or AML. And we know that clonal hematopoiesis, which is a pre-malignant state of clonal accumulation, that the mutations we see in that entity, whether you call it ARCH or CHIP or whatever you want to call it, are in the subset of genes that generally are epigenetic modifiers, P53, JAK2, but other mutations like NPM1 are not seen. And the implications of this are expanding, and there's a lot to figure out. But I think in the long term, what I'm most excited about is the idea that we can intercept disease before it occurs. And I think one of the things you'll see from the data I'll show you today and from other data from really great labs like Koichi Takahashi and from Kathy Smith and Adam Boddy and others using this platform is the concept that by doing clonal analysis, we may be able to actually get ahead of the game and actually move the chess piece in advance. And that's a concept I think that's really important. And so the fundamental question that we've been interested in the last few years is, if there are millions of people with precursor conditions to AML and other myeloid malignancies, and there are only tens to thousands of AML and 30,000 myeloid malignancies a year, it's clear that these early events are not gating or rate limiting to give you AML. And we want to understand, why is this common, and then why is this rare? And that's the fundamental thing I think that we really want to understand better, moving forward. And you could think of this in many ways. We can ask, what are the early events and what are they doing to increase clone fitness and expansion? We can think of this in the late stage about what do the events do at transformation. But I think most important is understanding the journey. What are the sequential events that occur to give you AML? What cell types did they occur in? Is there an order that matters? And how does the environment outside the cell or sort of milieu of cells change the fitness landscape and allow particular clones to emerge? And I'm just going to show you some data from our lab that gets at some of those issues, but we've got a long way to go for us to fundamentally figure that out. And so the way we think of AML is as a Jenga puzzle, that you have to think of AML as what are the building blocks that give you AML, and that's what I'll talk about today. But ultimately, we want to figure out, what are the therapeutic dependencies? What are the key blocks that if we pull them out, the AML can crash down? And if you ask me to give this talk in a year, I'll have a lot to say about that latter process. So perhaps next AML day, I can present some data on therapeutic targeting. And what I want to start with is, before we and others began using single-cell mutational profiling, it was evident to us from bulk sequencing that mutations in AML did not occur in random order, that there were some events like in DNMT3A, ASXL1, IDH, and TET2, I'll call that DTAI, if you will, are early events. And other events like FLT3 and RAS are late events. And the problem is that this data is population-based data, and it doesn't tell you about whether all these are occurring in the same clone or in the same order, or if there are different branches or even multiple branches occurring in patients at the same time. And so we thought that this data was incredibly illustratory, but it wasn't enough to really open the hood and ask, what does the wiring of AML look like? And so our long-term goal is to understand clonal evolution as a mechanism to decipher how myeloid transformation works. Can we better delineate the sequential acquisition of somatic mutations? Can we understand what the key events are that promote clonal expansion and myeloid transformation? Are there different routes or trajectories in AML in different patients or for different clones within an individual patient? And is AML a disease of a driving clone or a milieu of clones that are operating in an ecosystem? And ultimately, can we link all of this to phenotypes and to therapeutic dependencies? And so we think that many of these questions can be answered with single-cell profiling, but not with the typical sort of RNA-seq single-cell analysis you read about every week in Nature, which is incredibly useful, but doesn't let you track clonal evolution to the same granularities I'll show you about today. And so what Lindy Miles and Bobby Bowman did in my lab, working again with some of Hannah's colleagues at Mission Bio, was adopt a custom platform where we developed our own panel of genes similar to some of the off-the-shelf panels that you can get. We also were very willing to share what we did. We just thought there were some things we wanted to study that weren't on the existing panels when we began this work. You can see the genes there. And what we've done is single cell mutational analysis of about 146 patients with myeloid malignancies. The majority of them have AML, a mix of newly diagnosed and relapse refractory, a small number of myeloproliferative neoplasm patients, and a small number of patients with antecedent clonal hematopoiesis. And we've done a small number of samples in collaboration with Mission Bio comparing genotype to immunophenotype. And I'll show that towards the end. All of our data is available on our bioarchive, and I encourage you to look at that and then to tweet, email with questions to me and Lindy and Bobby. And I'll point out that Koichi Takahashi has really awesome data on bioarchive with complementary work. And actually Adam Abadi and Kathy Smith just had a bioarchive on some of those sort of, protein DNA methodologies. And I'd encourage you all to look at that because it really gives you a sense of where the state of the art is with this technology. And so what we did with our data was try to identify in every patient the different clones we see. As you can see here, this is what we all call, or what I call, a clonograph. It sort of gives you a frequency with the number of cells with each different genotype. And you could see already in this patient, we don't even see the antecedent initial clone that has whatever the first mutation is. So that already tells you that although this technology is great, we are not done by doing 150 samples at 10,000 cells per patient. I think it's going to be important to do larger number of cells and larger number of patients to really get the full clonal repertoire of a larger number of patients. And so the first thing we did was ask, does clonal diversity look different in different myeloid malignancies? And how does that compare, for example, to mutational burden or to diversity overall? And you can see the number of mutations increases from clonal hematopoiesis to AML, and you can see the different subtypes, those with signaling mutations, those with epigenetic mutations, or with epigenetic plus signaling mutations. So there is an increased number of mutations. You'll ask, why do the clonal hematopoiesis patients have 2 mutations and not 1? It's because we didn't look at patients that only have 1 mutation. There's really no question for us to do clonal architecture around a single mutation. But it makes the point that the mutational burden goes up, but the number of clones goes up more. And that suggests that although there's mutational acquisition, it's really not only going linearly, but it's branching out. And we think that's an important lesson. That in addition to mutations accumulating in one clone, we're actually seeing more clones being spit out. And that's probably critical to the mechanism of not just leukemic transformation, but malignant transformation more broadly. If you look at clonal hematopoiesis that is more than 1 mutation, the rule rather than the exception is the idea that we see oligoclonality. So when we see more than 1 mutation, they're in distinct clones, suggesting that clonal hematopoiesis pre-malignant states are not caused by clones that are evolving individually, but rather by one or multiple clones occurring in parallel. That is very different than the picture in acute myeloid leukemia. In AML, we see a few things that are really important. The first thing we do is that we see clones that accumulate mutations. So instead of parallel clones, we do see clones that are evolving compared to antecedent clones. The second thing is that some of these clones become dominant. And in fact, in 86% of our patients, we saw either 1 clone that was 30% or greater of the cells or 2. And what that means is that there are winner clones. And is that because those have better mutational combinations or repertoire? There are other sort of factors in those clones, stochasticity, or cell non-autonomous mechanisms where those clones suppressing other clones. I think these are all possible. And I don't think this data addresses that, but it gives you an observation, which now we can go back to the lab and functionally interrogate moving forward, meaning how much of it is due to the mutations enhancing the fitness compared to other clones versus other more complex interacting mechanisms. But the thing we wanted to begin to do is to measure diversity on a per sample basis. And so instead of just counting clones, we used a Shannon diversity index to measure clonal diversity. And you can see, again, as disease progresses, this goes up more than the number of clones that are mutations. So the fundamental thing here is that you're getting greater branching and diversity as disease progresses. You also see that although you have dominant clones, the dominant clone size is smaller in AML than in precursor states. And what that means is that AML, although it has clones that win, has many other very fit clones. And think of AML then as a milieu of many clones that are advantaged compared to wild type cells that are acting in a way either trying to win themselves or perhaps feeding off each other. And that, again, is a functional question that I think we in the field need to address. We then wanted to ask, all right, we take all this data, we have now a sense of who the dominant clones are versus subclones. Did different mutations have differential representation in the dominant clone versus other clones? You could see that some mutations like calreticulin and MPNs or NPM1 or IDH are in the dominant clone. When you have it, it's in the clone that's the winner. Other mutations like FLT3 and RAS are almost never in the winning clone. And then you have genes like TET2, which seem to have what I call a pleiotropic or joker-like role, where they can be either in the dominant clone or in a subclone. And that means that these mutations, some of them have more hardwired roles in pathogenesis, and others may have more pleiotropy or flexibility. And I think that's, again, a very interesting mechanistic question to dissect. But the real interesting question is not what do mutations do, but what do combinations of mutations do? And so on the left, we looked at cooperativity versus exclusivity in the bulk sequencing analysis. And on the right, on a per cell basis, you can see all kinds of matrices that tell you things about mutations that co-occur and don't co-occur. It's almost too complicated for a non-computationalist like me to really understand and get specific patterns. But we did look at some specific instances that taught us things. And one of the ones that taught us something were co-occurring epigenetic regulator mutations. So when you see DTAI mutations more than one, they occur in the same clone and they're in the dominant clone. And that means that it's not what does DNMT3A or TET2 do, something I've spent 10 years thinking about, but what does TET plus ASXL1 do, or DNMT3A plus IDH, as Peggy Goodell and others have tried to study. And this suggests that understanding how different epigenetic modifiers cooperatively corrupt or reorder the epigenetic state of hematopoietic stem progenitor cells is key to leukemogenesis. This is very different than for signaling mutations, which when they occur in different clones. You can see that JAK2, FLT3, RAS, generally with the exception of JAK2 and NRAS, occur in distinct clones. And you can see here that in this patient, you have FLT3, RAS, D835, ITDs, PTBN11, virtually all occurring in distinct clones, suggesting that what you're getting is a milieu of subclones or daughter clones, each of which has its own signaling effector that's contributing to its enhanced fitness. And in fact, if you look at signaling effectors versus epigenetic regulators, you can see different co-occurring patterns. So for example, JAK2 mutations only occur in IDH-only clones. JAK2 plus RAS can occur in the double mutant DNMT3 IDH clones, and the FLT3 mutations either occur in IDH1 clones or in DNMT3 IDH2 clones. So again, not understanding what do these individual mutations do, but why do certain sort of complemented mutations, boom, give cells enhanced fitness. The data, though, to this point tells you about who is with who. It doesn't tell you about who with who enhances fitness compared to others. And so to do that, we use the Markov decision model to actually map trajectories from normal stem cells to fully transformed cells, and then look not only at the order, but at the relative fitness. So if you look at this patient here, it's a DNMT3A mutation, still a small clone, then an IDH mutation, so it's a double mutant, still small clone, but boom, that clone picks up NPM1 and now it's expanded. And you can see here in this patient, IDH FLT3 NPM1 has massive clonal expansion compared to IDH NPM1 and RAS. And this suggests that these particular triads of mutations are the switch where you get this massive increase in fitness to the detriment of other clones. And so we asked then, do different mutations have different roles, either in initiation versus clonal expansion? And you really see 4 different patterns. JAK, IDH, DNMT3A, when they occur, they're early events. FLT3 and RAS pathway mutations are late events. NPM1 and ASXL1 you can think of as being not first, but often the second person to the party. They're the thing that defines the disease and is in every cell in the dominant clone, but is not the initiating mutation. And then you have TET2, which, as I said earlier, is pleiotropic, which has the most flexible role. It can be initiating or acquired and transformation or in subclone. And so we then ask, can we experimentally find those early events? Because if we say, well, in this patient, JAK2 is the first event, do we see JAK2 only cells? And the answer is, in blue, we do sometimes, and in gray, and sometimes we don't. But if you look at when we do in blue, it matches our predictions quite well. But the large number of gray suggests that what we're missing in many cases is due to limitations in sequencing. And so I really believe that we're going to need 1 to 2 logs greater in sequencing. It's a little bit like asking, do we have enough COVID testing? We need a lot more sampling of many more cells, the same way we need a lot more sampling of patients in the community. And I think the good news for all of us is that this technology is going to get us there. And I'm very excited in the next year or two to see what all of us as a field do using this technology to mine a larger number of cells and a larger number of samples. There are other things that we observe. For example, for DNMT3A, the canonical RAA2 mutation is almost never seen as a single mutant clone in our data set. And we don't know if it's because that's different than other 3A mutations. Is that because those mutations don't initiate, or because once you get it, boom, they take off and you're very hard to find those? I think without deeper sequencing, we won't be able to delineate that. And that's going to be a really fun question to think about. But when you begin to think about mutational combinations, you can actually start to look and ask, do specific combinations which co-occur have greater impact in enhancing clonal fitness than other mutations? So these 3 combinations, DNMT, IDH 1 and 2, or NPM1 FLT3, massively increase clonal number and clone size and are cooperative. But NPM1 and RAS, they are in the same clone. I'm not saying they're deleterious to each other, but they're not synergistic. And I think this, if one goes back and asks about mechanisms of cooperativity, this could be additive and these could be synergistic, or this could be neutral to negligible and these could be synergistic. And that's going to be really fun to sort of figure out. And I'll talk a little bit about at the end about where we're going to go moving forward. But the other cool thing we can do with this technology is begin to look at samples at more than one time point. So we've just scratched the surface on this. And this is a patient who had MPN and red and transformed to AML. And you could see when they developed AML, they went from having these clones here that were all CALR ASXL1 mutant to having a CALR ASXL1 IDH mutant clone. But that clone was already there pre-therapy. So again, imagine if you could detect it, monitor it, or even treat it. And so that's why this idea of sort of interception comes from, ultra-sensitive single cell sequencing. We can also look at patients during therapy with targeted therapies. This is a patient pre-post FLT3 inhibitor therapy who went from having a FLT3 mutant clone to having RAS mutant clones. And this is very similar data to previously published data from Sasha Pearl and Kathy Smith showing that the RAS clones can emerge during FLT3 inhibitor therapy. The difference here is that what we found in this patient and in this patient is even more illustratory, is that the RAS clones were actually in FLT3 wild type cells. And that means there may be 2 routes to transformation from a FLT3 mutant disease treated with a FLT3 inhibitor, where the FLT3 clone can acquire the RAS mutation, or in some patients, there's an antecedent pre-FLT3 clone that acquires RAS. And that may have very different implications for how you monitor and ultimately treat patients. And we need to get beyond these early anecdotes to larger numbers of samples. And that'll be fun to think about moving forward. And so the challenge, though, is that all of this is just genotype. It doesn't tell you anything about what the cell is and what it does. And so obviously it'd be fun to link DNA to things like cell surface immunophenotype, RNA gene expression. And so what we did, thanks to, again, our colleagues at Mission Bio out of my body, is begin to use genotype linked to immunophenotype, very similar to the site-seq technology, where you can use the conjugated antibodies from BioLegend, which let you basically match up immunophenotype for surface molecules to genotype. And so this is what the data looks like. You get all these different clones in this AML patient. The wild type cells are all CD3 positive, so those are the T cells, but all these different clones have different immunophenotypes, and we can match up genotype to phenotype. And so what can you do with this technology? And so this is a good example of a patient with a DNMT3A mutation. In this patient, you have a C451D, has a R379L frameshift, and this mutation is in the myeloid cells. You can see it clearly in the CD11B cells. And you can see it as well in the T cells, but not in the B cells, making the point that this mutation is represented in both. But in this patient who has 2 different DNMT3A mutations, you see very different answers. You have the R882, which is in the myeloid cells, but not really in the T cells, and it's to some degree in the B cells, and this R635Q, which is in the T cells, but not the myeloid and B cells. So you can basically map out what all of these mutations are in different hematopoietic compartments. And then we went back to our AML data, and what we're able to do is to show that in different AML patients, we can identify subclones like this patient that had all these different clones, but the RAS mutant clones had very specific immunophenotypes, and in each case, the RAS clones were associated with the increased expression of CD11b, suggesting that the RAS mutant subclones have a cell surface repertoire different from others. So imagine you had a conjugated toxin-mediated antibody or cellular therapy or something targeting a cell surface molecule. It could be that genomics could be used to pick out the right subpopulation, the right cellular therapy, and what else to combine it with. And so that may be somewhat forward-thinking, but that's the path we think it'll be important to go down. And the important thing here is that in 8 of the 14 patients where we did this, the dominant clone had a different immunophenotype than the subclones. And so if you want to target the subclone surface, it may be very different than targeting the dominant clone. So you couldn't get this from bulk flow cytometric profiling of the dominant clone. You really need to be able to look in the dominant clone and in those rare subclones. And we can begin to look at not only to what a mutations do to surface expression, but combinations of mutations. You can see, for example, that IDH GNMT3I double mutant cells are 34 positive, whereas NRAS IDH1 cells are CD11b positive. And early days here, we need to get beyond 7 markers. We think you got to get to 30 or 40. You want to get from not only DNA to protein, but DNA to RNA. There's going to be a lot of fun in the next few years to begin to do clone-specific phenotypic and transcriptional analysis. So in summary, before I just give you a sort of a few forward speaking statements, I really think that single cell sequencing allows you to look at AML under the hood to really understand the molecular circuitry on a per cell basis. Clonal diversity increases disease progression, and dominant clone size decreases disease progression. We can see how specific mutations and combinations contribute to initiation, clonal representation, and propagation. And combining this with antibodies gives us our first look at clone genotype to clone phenotype. Lots to do moving forward. Link this to clinical outcome and therapeutic response. Look at serial samples at reasonable numbers and integrate RNA and additional antibody tags. So I think there's going to be a lot of fun to be had in the next few years. And the fun thing for my lab is that what we're going to do is use this data as a blueprint to go back and understand AML pathogenesis. And so if we can delineate the order of mutations, we can go back to the lab and model it. And so this is data from Lindy, where she developed AML models, where she either inactivated TET2 and then activated NPM1 or vice versa. And only if you do the order the way it occurs in the humans in the right clones, where it's TET2 followed by NPM1, do you get a fully penetrant AML phenotype. And again, just to give you a teaser for next year's AML Day, what we're really doing in the lab now is developing the mechanistic tools to actually functionalize and study and model the order and milieu of clonal events that we see in AML patients. And we'll have a lot to say to this moving forward. And more important than having me talk about it will be to have people like Bobby and Lindy talk about it, who are really sort of pioneering these efforts. So with that, I'll stop. I'll just acknowledge we've had a lot of help again from our colleagues at Mission Bio, including Robert, Ike, and Pedro, and many others. Bobby and Lindy have done all of this work with lots of help from Aaron. Mike Wartz has helped with some of the work moving forward. A lot of help from Tiff and Nicole in the lab. Richard, Mithat, Mike, Amit, Kelly, who you heard about giving a talk to the ACR, Agnes, have been critical. And we also want to thank our outside collaborators, Jason Butler, who's helped us with our functional studies, Sarah Meyer and Martin Carroll, who've been great collaborators with us on single cell sequencing, Yanis Aifantis, who's one of my longest collaborators, and Todd Druley, who's really helping us compare Mission Bio single cell to other ultra-sensitive sequencing technologies. We'll have a lot more to say about that. Thanks again for having me. And if we have time, which I think we do, I'm happy to take questions. Hannah, I'm gonna stop my show and hand the ball back to you.
Great. Thank you so much, Ross. That was an amazing presentation. So we do have a couple of questions here. So TET2 and IDH1/2 mutations were thought to be mutually exclusive. Is there something specific about the type of TET2 mutations that coexist with IDH?
I think that there's a lot we don't understand about that. And I think that fundamentally, you know, as some of you know who think about IDH and TET, you know, the IDH oncoprotein inhibits many other enzymes, and it could be that the other non-TET2 dependent enzymes that IDH affects allows it to cooperate with some mutations different than TET2, and we think there's a lot to figure out there.
Okay. Okay. I think there's a lot of interest in using technologies like this to do minimal residual disease. What's your thought for the application of this there?
I think it's great. And I think Ravi Majeti just had a paper on blood advances where he did some of that. The challenge with minimal residual disease is that it's rare. And so you're doing single cell profiling of a rare population. And so the question you have to ask yourself is, do you want to go ultra deep with single cell when 98% of your cells are wild type? And so I think the ideal strategy will be to come out an enrichment strategy for MRD with single cell profiling, because you don't want to waste all of your genomic real estate. If only 0.1% of the cells are leukemic, do you want to sequence the other 99.9 over and over again when what you really care about? So I actually think that one has to think about that. Now, one could also think of, for example, of using AbSeq as a way to do that, but you still have the challenge that you're sequencing wild type cells. So as capture rates get better, it's greater, but we believe that in 2020, the ideal strategy would be to combine some sort of enrichment strategy followed by MRD analysis. That's just my opinion.
Okay, great. Aren't some of these somatic mutations also present in older individuals and not necessarily specific to AML?
Yeah, as I said earlier, it's the phenomenon of clonal hematopoiesis. And what this means, and some of the analysis we did were in these older folks who don't have leukemia. And so the other interesting question, which I think AbSeq or DNA protein analysis will let you do, is to ask, do mutations and their contribution to different lineages differ in an actual overt disease state versus this older precursor state? We don't know the answer yet. Is 3A and the same distribution of cell types in a pre-malignant state as in a malignant state? It's a really interesting question.
Okay. Can you explain again why the dominant clone size goes down with transformation and presentation of AML?
I don't think I know for certain. I think the hypotheses we have is that there's been enough clonal evolution that there is more than one clone that has a massive fitness advantage, or alternatively that the winning clone is secreting factors that not only enhance its own fitness, but perhaps other clones, and perhaps there's crosstalk. These are interesting questions that need to be studied in more granularity.
Okay, great. Just a couple more questions here. So I think you talked a little bit about it, but can you maybe articulate again how you foresee how this type of information from single cell clonality will be able to inform therapy decisions? Like, would you have to target all of the mutations? Is there an order of therapy that could be used? Some dynamic therapies people have thought about?
I think that's a really interesting question, and there's a lot to think about and unpack there. What I'd say is a couple of things. The first is you want to start by taking mutations that are rare but have targetable sort of proteins like IDH or FLT3 and start asking, if we targeted them before we normally would with bulk sequencing, does it make a difference? And does sequence and order matter? So if we can write the rules, then we can design more comprehensive strategies. So I think there's still a lot to do there. I also think, to be blunt, that we have to learn from the antiviral world, whether it's Hep C or HIV, about how to use combination strategies. That oncology, will only ultimately be successful when we develop combinations like this, the same way people have thought about virology and the different routes a virus can take. And so I'm hoping that we don't have to write this book ab initio, if you will.
Makes sense. So maybe, connecting it back to sort of AML Awareness Day and your clinical practice, obviously it's changed quite a bit in the last couple of months. Have you seen any patients with AML infected with COVID? How is your clinical practice going these days?
I go on service in a week, so ask me then. But what I would say,
Okay.
is that at most centers in New York, including our own, we have seen patients who have COVID-19 disease with a broad spectrum of malignancies, including heme malignancies. We are at Sloan screening people who are going to get therapy for leukemia for COVID, so we know. And in some cases, if they're positive, we follow them to make sure they're stable before we treat them. I think there's early data from the group in Wuhan, very limited numbers, that's on one of the preprint servers, suggesting that outcomes are significantly worse with heme malignancies and AML with COVID disease than other cancers or patients without cancer. But we're going to need a lot more data in the next couple months. But I think it's a really scary moment for all of us providers, patients, their families, because there's a lot of unknowns, and we have to do the best we can. And you can't delay AML therapy forever. And so the answer, unlike maybe for other disease states where you have more time, there's some difficult decisions. And I give the patients and their families and their doctors a lot of credit for the courage to make decisions where there is no right or wrong answer.
Thanks for that. So you talked about, trying to stay positive and productive, I think, during this really difficult time. How are you staying connected to your community, and how are you operating with your lab? I'm sure lab meetings look a little bit different these days.
Yeah, so, everyone labs do it different. We do a check-in on Monday morning, just that I think everybody can just see each other's face and see if there's anything logistical or to share. We do a journal club every week. Most of the time it's based on science. We did do a careful academic review of Tiger King last week. Which I think is more fun than academic, but I'll leave that to the lab. And we do a lab meeting each week, which is either going over projects, and when we're get to the point where there's no new data that we haven't looked at, we'll start talking about strategic plans. I'm listening to lectures like this. There's a great series, unfortunately, going on right now called the Blood and Bone series, which I've spoken in, the DFCI and MSK. DFCI is a series at noon, 3 days a week. MSK's got a series on Mondays and Wednesdays at 4:30. Craig Thompson's speaking tomorrow at 4:30. So I'm sort of soaking some of that in. And then the last thing is I'm not wallowing when I'm not getting everything done I wish I can get done. I'm doing the best I can. I think, whether it's, cooking dinner more with my family or, watching, my wife and I are watching Unorthodox on Netflix this week, we're like, which probably if I was traveling and going to dinners, we would have done separately on a plane. I think it's more fun to do some of these things together. So I know that's like very small things that one can take advantage of in a very challenging landscape. But if one focuses on that and not on the sort of other aspects, I think we'll all get through this a little bit better. And I think for those of us who are affected personally by the disease, which is almost everybody, I know that everything I just said, rings somewhat hollow in the circumstance where someone around you or yourself are impacted. And obviously that's where all of our thoughts are.
Well, with that, Ross, we appreciate you being here and taking the time, despite the circumstances, and on AML World Awareness Day. We hope to see you next year here with some other news about the research and the work that you're doing. And again, we appreciate it. Thank you to all of you for joining, and we hope that you all stay safe and healthy.
Thank you.
Thanks. Bye-bye.
Bye.